The previous presentation showed how mass spectrometers work, but why is the mass of a peptide important? As mentioned at the start of the last presentation, a unique chemical structure can have a unique mass that is diagnostic of the molecule or structure. Peptides can have a unique sequence of amino acids, and thus a unique structure, such as this one for glufibrinoleptide B. The proteins also have unique sequences, and when cut up by specific proteases such as trypsin, which cuts after lysine and arginine, measuring the masses of the produced peptides can identify a protein, such as myoglobin here. This method is still used in some labs to identify proteins separated by 2D gels, where a single protein spot is digested to peptides, and the peptides are measured by Link2 mass spectrometry. The limitation of this technique is twofold. Firstly, if more than one protein is present, data interpretation can be a little difficult. The other problem is with peptides that have the same combination of amino acids, but different order to their sequence of the amino acids, such as in this example, where the sequence of amino acids has been reversed. Measuring the intact mass of these peptides would not reveal their identity. The solution to this problem, and now the basis for identifying peptides using mass spectrometers, is to fragment or break the peptides into smaller pieces and measure the mass of the fragments. If we take the forward peptide sequence and we break the peptide between the 5th and 6th amino acid, we get two fragments of 536 and 1059 Daltons. If we take the reverse peptide sequence and break it again between the 5th and 6th amino acid, we get two different sized fragments of 626 and 969 Daltons. So even if these two peptides were mixed together and the mass spectrometer selected the mass of 1570, the mass of both intact peptides, the different masses would tell you that more than one peptide was there. To see how the mass spectrometer does this in practice, we return to part of the quad-top animation. As mentioned in the last presentation, the quadrupole 1 can be used to select only one ion to the exclusion of all other ions being sprayed in. These ions then get transmitted to a collision cell filled with nitrogen gas, where fragments are produced by collision with those nitrogen gas molecules. So the mass spectrometer is continuously executing a sequence of scanning for intact peptide ions, selecting one of those ions, fragmenting it, and measure the mass of the fragment, and then repeating that. In the scan for intact peptide ions, a mass range of 350 to 1500 Daltons is typically scanned, but this spectra for glu-fib has been zoomed in. Using electrospray, multiply charged ions are created, and peptides having multiple amines that can be protonated are at least doubly charged. As the mass spectrometer measures mass divided by charge ratios, the difference in mass between the C12 and the C13 isotope is half a Dalton for a doubly charged ion. This is used to select ions for fragmentation rather than have the instrument waste time fragmenting non-peptide ions. The molecules of the C12 isotope of glu-fib are then selected by Q1 and transmitted to the collision cell, where collisions with nitrogen gas molecules cause fragmentation. Thousands of molecules of the same peptide are present, and different molecules fragment through different bonds, thus producing different mass fragments. When these are measured, this is the resulting spectra. This spectra has been annotated to show the sequence of the fragments, but the mass spectrometer does not know the sequence. The sequence can be determined afterwards by calculating the difference in mass between adjacent fragment peaks and seeing whether the mass corresponds to an amino acid mass. For example, the mass difference between the 1285 Dalton peak and the 1171 Dalton peak is 114 Daltons, which is the mass of the amino acid asparagine. When we discuss the fragments of a peptide generated by a mass spectrometer, we use a nomenclature to describe through which bond a peptide molecule is broken to generate a fragment. Peptides mainly break through the peptide bond, in this case joining the carboxyl group of the first or N-terminal amino acid in the chain to the amine of a second amino acid. When the molecule is broken through this bond, the N-terminal end is called the B ion, while the C-terminal end is called the Y ion. We're going to use this example that comes from Karen Jonch's slides from the University of Colorado in Denver. We're going to manually interpret a spectrum generated by an ion trap. The mass that was measured by the ion trap is 1449 Daltons, and it was doubly charged. We're going to start by looking at the dominant peaks that are below the mass of that precursor ion. So we need to look for ions that are between 57, the mass of glycine, and 186 units, the mass of phenylalanine, below the mass of the ion detected. So here's our spectra, and the first ion in the spectra is 1337.5 Daltons. If we subtract that from 1450, we get the mass of either leucine or isoleucine. These two amino acids are isobaric. They cannot be distinguished from one another, so we designate this as LXX. The next ion down is at 1276. Now, this ion is less than 57 away from 1337, so it can't be the next amino acid in the series from the N-terminal. Therefore, it is most likely the first amino acid from the C-terminal of the peptide. Because this is a tryptic peptide, the C-terminal amino acid is going to be either lysine or arginine. So if we subtract 1276 from 1450, and we include the mass of a water molecule, which is induced during fragmentation, we get 156, or the mass of arginine. Therefore, we can assign arginine to our C-terminal. The next one down is at 1238. If we assume that because the mass difference is too close to 1276, it's most likely the next amino acid down from the leucine or isoleucine. If we subtract, we get 99, the mass of valine, so we assign that next. The 1219 ion is 57 Daltons away from 1276, and therefore it's the mass of glycine. We can continue down looking at the masses of the ions and working out whether or not they should belong to a separate series. At the moment, what we're determining is a little bit speculative, because we're not absolutely certain whether or not the combination is correct, but the math makes it correct. What we need is we need to find ions from the other end of the sequence, small mass ions from the other fragments, to complement our higher mass ions. So with ion trap data, we normally don't see ions below about 200 Daltons, so we need to start looking for the ion that would reflect leucine and valine joined together, or B2. If we add together the mass of leucine and valine, we get 113 and a hydrogen atom and 99, which equals 213, which is present in our spectra. If we then look at what we need for another valine, leucine, valine, valine, we need a mass of 312, which is also present in our spectra. Therefore, we can start walking up our mass range looking for ions that complement the C. When we work our way through the entire spectra, we should be able to assign all of the major mass ion peaks in the spectra. What we've described here is the determination of the sequence of a peptide by de novo means, or without any prior information. Once the number of fragmentation spectra exceeds 10, de novo peptide sequencing becomes a little impractical. In another presentation, we will discuss how search algorithms can use databases of protein sequences to determine the sequence of a peptide in a spectra.